Actually, I'm very delighted to actually have the possibility to present to this very interesting meeting organized by the Patients Association Mitocon. So today I'd like just to give you an overview of the recent work we have been doing in my lab in a full collaboration with the Valerio Carellis and Valeria Tiranti's group. So basically my lab in the last decade was fully committed to try to further enhance and elaborate the technologies for generating human individual specific stem cells, known as iPS cells or iPSCs in short. These iPSCs are actually generated by the direct conversion of skin and blood somatic cells from adult individuals. And these stem cells can be maintained in vitro and then can be differentiated in any other somatic cell type. And this is particularly useful for those cell types which are hardly accessible in the human body, like neurons, hepatocytes, or beta cells of the pancreas. So in this case, we have an in vitro model which generate individual specific cells which can be affected in a specific disorder, and we can use these affected human cells to study the molecular pathophysiological mechanism, or even to initially try the innovative therapies directly into the patient affected somatic cells. So over the years, we have learned how to generate iPS cells from really a tiny amount of blood samples, few milliliters. And we actually developed best practice for actually generating high quality iPS cell culture, and then to actually differentiate them in a very efficiently into either neuroprogenitor cells, which can then be differentiating in pure population of either neurons or astrocyte or microglia. And these actually cells are fully endowed with all the functional properties which can actually are found directly in the body. So this is really a formidable experimental in vitro system to actually study the diseases, especially neurological and neurodegenerative diseases, which is the main focus of my lab. And this is the only way to have access to human neurons affected by this disorder to study and to actually try novel treatments. So in particular, my lab has been developing iPSCs from genetic and sporadic forms of Parkinson's disease, in particular from those genetic forms caused by a mutation in a dozen of genes, which many of which actually encode for regulators of mitochondrial biology. So mitochondrial dysfunction seems to be strongly correlated with the onset and progression of Parkinson's disease. However, the cellular processes which are mostly affecting mitochondria by mitochondrial dysfunction and leads to neurodegeneration are not still completely understood. And this is true for Parkinson's disease, which is the second most common neurodegenerative disorders, leading to strong, severe motor impairments due to the specific degeneration of dopaminergic neurons, which are localized in small nuclei in the brain, which are known as substantia nigra. So why these neurons are specifically vulnerable to this disease is not fully understood. So on this view, I was particularly intrigued when actually the Valerio Carelli's team in Bologna described for the first time two large families with several members suffering of frank Parkinsonism who actually harbor an heterozygote missense mutations for the OPA1 gene. So this was the first time that it was clearly described that OPA1 gene deficiency was actually associated with a frank and clear clinical Parkinson disease manifestations. So I was very intrigued by that. So I team up with Valerio Carelli's and Valeria Tiranti's group, try to better understand this correlation. So OPA1 is encoding for a very critical mitochondrial GTPase with pleiotropic function in mitochondrial biology. The work by Lucas Corrano's lab and others worldwide have actually nicely showed that OPA1 is fundamental for promoting mitochondrial fusion to actually maintain stability of mitochondrial crystal and also to counteract mitochondrial dependent cell death. Moreover, we know very well that OPA1 gene mutation are causing pure or syndromic forms of dominant optic atrophy, DOA. However, whether this patient might actually suffer also of neurological disorders early in youth or in elderly is not very well known, and still, you know, there are sporadic observation, and so therefore we were very curious to better understand the cellular basis of this correlation. So to do that, we actually team up together, and Angelo Iannelli, a research scientist in my lab, who did most of the work I'm presenting to you, actually generated iPSCs from 2 patients, one for each of the 2 families described by the Carelli's group. Then we generate also iPSCs from 2 normal donors, and we also use 1 patient cell line to actually reintroduce in a stable way an OPA1 gene functional copy in order to restore the overall OPA1 gene expression level. So we use this line as a line to actually see the rescue of the phenotype. So Angelo differentiated each of these iPS cell line into neuroprogenitor cells, in short NPC. This is an homogeneous population of proliferative neural cells, and OPA1 patient NPC show a clear, significant fragmentation of the mitochondrial network, as you can see here in these 2 representative examples. And this fragmentation was fully rescued in the patient cells, which restore OPA1 gene expression levels. Compatible with that, Valeria Tiranti's lab actually showed that OPA1 patient NPCs have a strong reduction in oxygen consumption, coupled with an impairment in production of ATP, and we showed that this impairment was specifically coming from an impairment in complex 1. So overall indicating that there was a dysfunctional oxidative phosphorylation activity in these NPC. Angelo also noted that OPA1 patient cells have tremendous high levels of radical oxygen species. And particularly interestingly, the reintroduction of the OPA1 gene functional copy was sufficient to actually restore normal level of ROS in these cells. Angelo then differentiated these patient NPCs into fully mature neurons. All these red cells you see are neurons, are human neurons. And while neurons from normal donors can be maintained in vitro for really many weeks, up to 4 months, we had much more difficulties to actually maintain a good population of neurons from the OPA patients. So indicating that these neurons have an increased tendency to actually die or to be lost in vitro, in the vitro culture system we are using. And interestingly, we could rescue this inability to survive of the patient cells by supplementing the medium with a specific inhibitor of necroptosis. Necroptosis is a necrosis-regulated cell death modality, which depends by the overactivation of the RIP kinases. And indeed, we found that RIP kinases are actually overactivated in OPA1 mutant cells. And we found this also in post-mortem Parkinson disease brain tissue. However, what Angelo noted in OPA1 patient neurons was that in the neurites of these neurons, there was a significant loss of mitochondria. However, we recognize that the differentiation of in vitro of stem cells into neurons generate a culture of disordered and not well, a disorganized ensemble of neurons. And this is dramatically different from what we experience and what is actually are the neurons organized in the brain, where all the connection are topographically organized, connecting specific neuronal cell type between each other. While in the culture, as you can see here, the neurons can contact any other surrounding neuron. So there is no any modeling of the neuronal circuits which are topographically organized in the human brain. So try to solve these important caveats of the in vitro cultures we have in the lab. We decided to try to model specifically and to reconstitute in the dish the connection between the dopaminergic neurons and the basal ganglia neurons, which is called Nigro-striatal pathway, which degenerates specifically in Parkinson's disease. So for this, we team up with Marco Rasponis at the Politecnico di Milano, who generate for us a microfluidic platform, which is organized in two lateral chambers here, where we could plate the iPS-derived dopaminergic neurons, and in the other lateral chamber, we plate the basal ganglia neurons, let them to differentiate, and these neurons are obliged to send their axons in this microchannel up to extend to a central chamber that we call the synaptic chamber, where supposedly the synaptic contacts are formed. So this is an example of how it works. So here you see in green, GFP positive, iPS human dopaminergic neurons. The soma are in the lateral chamber, and they send their axons in these microchannels up to the central chamber. And in the same way, the red colored basal ganglia neurons differentiated from human iPS cells can send their axons into this central chamber. And we prove that in the central chamber, there is a specific staining for synapse-specific markers like common synapsin, indicating effectively the generation of synaptic contacts spatially organized in this central chamber. So I think we use this model as a model that reconstitutes specifically a brain circuit exactly with the neuronal cell type, which are fundamental in the disease pathology. So this model allowed us actually to really image and to perform advanced imaging specifically in the dopaminergic OPA1 mutant axons. This is just a representative imaging that I point with my arrow. As you can see, these are the mitochondria present generally in normal human axons, while here are the amount of mitochondria which are strongly reduced in numbers in OPA1 patient dopaminergic axon. And sometimes these mitochondria are also clustered in abnormal configurations. So when we actually did dynamic imaging of these mitochondria along specifically the dopaminergic axons, we have evidence that these mitochondria has a failure or they present a reduced mobility along the dopaminergic axons. So indicating that not only we have a strong reduction in the mitochondrial mass in the axons, but they also have a tendency to be less motile with respect to the controls. Is this failure in mitochondrial presence in the axons leading to a defect in synapses? With this reconstituted circuit in vitro, we could actually answer to this question. So we image the amount of synaptic contacts in the central chamber. All these green dots represent a specific synapse, and we could actually find that even that OPA1 mutant axons produce a far reduced number of synapses. And very interestingly, this decrease in the number of synapses was dependent by the time. So more time we kept in culture these neurons in the microfluidic system, less synaptic contacts we could actually observe in the synaptic chambers. So all these data indicated us a model which suggests that in OPA1 patient dopaminergic neurons, the amount of mitochondria in the axons is strongly reduced. And this leads to actually a loss of synaptic contacts due to the lack of mitochondria to sustain the local energy, which is necessary to maintain the high metabolism of synapses. So the idea is that the degeneration which might occur due to the OPA1 mutation is caused by a dying back mechanism. So the first structure which are dying are the synaptic contacts between dopaminergic and basal ganglia neurons. And then the dopaminergic neurons is found itself with less contact with the neuronal network surrounding, and therefore is less excited. And this leads the neuron then to be fated, to die due to a lack of interconnection with the neuronal network. So we got also interested to another mutation which can actually cause Parkinsonism, and these are mutations in POL gamma. POL gamma mutation can actually lead to a wide variety of neurological symptoms, among which, especially during elderly, Parkinsonism is actually quite well associated with this type of mutation in POL gamma. Indeed, there is a clear correlation between mitochondrial DNA homeostasis in actually ensuring the survival of the Nigro-striatal pathway. There is controversy on this point because I think we still need to understand important concept in this correlation. And we think that iPSL technology might actually help to actually clarify this issue. So again, thanks to collaboration with Valerio Carelli and Valeria Tiranti's team, we actually all agree that this was actually an interesting topic to study with iPSL technology. So Valerio has a patient with an homozygote mutation. This mutation is in the linker domain of POLG, is P648R. And this patient actually has a severe Parkinsonism together with other neurological symptoms, of course. And we derive iPSCs from this patient and the second patient from the Valeria's that Valeria actually provided to us. And Angelo generated the iPSCs from these two patients. Now I'm showing only the iPS cells from this particular patient. Then we had iPSCs from two different neuronal donors. And in this case, Angelo actually corrected the point mutation in one of the two alleles by CRISPR-Cas gene editing, generating a patient-specific isogenic control. So the patient neuroprogenitor cells actually grow much slower, and they lose proliferation after a short while, differently from NPC derived from normal or gene-corrected cells. Mitochondrial network in this case is normal. But more importantly, we also observe that these POLG patient NPC have a reduced mitochondrial DNA copy numbers that was really consistent in all the samples that have been tested in Valerio Carelli's group. So again, we all together collaborate in actually provide evidence that POLG patient NPCs have reduced protein levels of mitochondrial encoded protein like OX2 or complex 4, or in general of complex 1 proteins. And this is associated with a significant impairment in oxygen consumption and ATP production, again indicating an impairment in oxidative phosphorylation. However, actually, we got really surprised when actually Angelo showed that POLG mutant NPCs have a complete failure in differentiating in post-mitotic neurons. All these red dots are post-mitotic neurons. So you see that differentiating iPS cells from normal donors or gene-corrected patient cells, we obtain many neurons. While in the differentiation plate of the POL-G patient NPC, there was a completely lack of neurons. This was really astonishing, as was never been described before in the literature. So we wonder how this might actually happen, that these mutant NPC have lost completely the potential to generate neurons. So we tried to rescue this phenotype with different molecules or conditions, and the only way we were able to fully rescue the neurogenic potential of the POL-G patient NPC was by doing the differentiation in an hypoxic condition. So in low oxygen tension in a lab, in the laboratory, which is 5% of oxygen tension. This condition fully restored the ability of differentiating neurons from the POL-G patient NPCs. Hypoxia condition generally induce the activation and stabilization of a transcription factor, which is the hypoxia-related IF1 alpha transcription factor. So we tested whether activation of IF1 alpha was sufficient to restore the neurogenic potential of POL-G patient NPCs. And indeed, this is the case. So we could actually obtain, fully restore the neuronal differentiation in POL-G patient NPCs by re-expressing only the transcription factor IF1 alpha, or simply stabilizing the IF1 alpha protein in high oxygen condition with a specific drug. So in both conditions, we can restore neuronal generation, and this was associated with an increased expression level of glycolytic genes and antioxidant genes. So this IF1 alpha reactivation is really necessary to rescue the defects in neurogenesis of the POL-G mutant NPCs. But we then ask a following question. What happens to the amount of mitochondrial DNA in these restored POL-G patient neurons? And to our surprise, we found that the total amount of mitochondrial DNA copy was actually restored from this level in the patient to this level after actually exposure to IF1 alpha expression or stabilization by the small molecule. So indicating that activation of this hypoxia condition are also capable of stimulating mitochondrial DNA replication. And this was confirmed by the restoration of some of the levels of mitochondrial encoded proteins, and was associated with an increased expression levels of protein strongly associated with mitochondrial DNA replication, like TFAM, Twinkle, and SSBP1. So all these three proteins, which are core in the molecular machinery controlling mitochondrial DNA replication, are strongly increased in this condition. And probably these are why POL-G mutant NPCs restore their ability to actually differentiate into neurons. However, in our studies, we did not find any evidence of a direct activation of IF1 alpha of these particular genes. So we don't think that there is a direct activation of these genes by IF1 alpha, while what we actually came across was the finding that in the POL-G patient neurons restored by IF1 alpha overexpression, we had very high level of lactate dehydrogenase, which was present inside the nucleus. This is not the first time that has been described. This was actually described before in the literature, showing that high level of ROS induce an increase in lactate dehydrogenase, which is also observed in the nucleus. And the nuclear fraction of LDHA is actually binding to the H3K79 methyltransferase L1. This is the methyltransferase that methylates the DNA and stimulate transcriptional activation. So where there is an enrichment of this methyltransferase, gene transcription is stimulated. So in our POLG patient neurons stimulated with IF1 alpha, we found increased level of LDHA and increased level of H3K79 3-methylation, indicating a general increase of DOT1L activity. But most importantly, we found a strong enrichment of DOT1L in the proximal promoter of the TFAM gene. So suggesting that the TFAM increased expression levels in the mutant neurons might actually be also due to a strong enrichment of the activity of this methyltransferase. So indicating that the rescue that we have observed is depending by an epigenetic mechanism mediated by activation of the hypoxia-specific IF1 alpha transcription factor. So I think this is my last slide before going to actually the acknowledgement. This is the lab working now in these conditions. It's not easy at all, and I really am very thankful to these really people that really work in this difficult condition. And I already mentioned Angelo Iannelli, and all the work I've shown is a full collaboration with Valeria Tiranti and Valerio Carlelli's lab. And we hope to actually have been the possibility to continue to explore these topics with iPS cell technology. So thank you. I stop here and I thanks for your attention.